We're going to switch it up here now and begin our discussion on microbial anabolism, which is the formation of molecules from smaller molecules, or storage, essentially. So, one example of an anabolic pathway happens in organisms that are called phototrophs. Phototrophic microorganisms are going to derive their energy from light specifically. And this might ring a bell. You might say, well, I've heard of those folks before, and they're typically going to be photosynthetic. And photo, of course, means light. So, how these processes work is by the reduction and the incorporation of carbon dioxide from the environment following the harvesting of energy from the sun in the form of light. And this is done through the Calvin cycle. Most phototrophs are also considered autotrophs because they use carbon dioxide as a sole carbon source. So they're not going to be using organic forms of carbon, but rather solely carbon dioxide as that initial building block for everything that they're going to create. It's funny because when we think of photosynthesis, we automatically think of plants, but over half of the photosynthesis on Earth is actually performed by microbes. This little table here just simply shows some examples of eukaryotes and prokaryotes that perform photosynthesis, and this is not a complete list, obviously, but you can see that some of these different prokaryotic organisms we've already talked about when we looked at our phylogenetic survey a long time ago. So when we look at photosynthesis in eukaryotic cells, we know that the process is going to occur in organellar structures called chloroplasts. These systems are membrane bound, and even within the membrane bound chloroplast, there are even more membrane bound internal structures called thylakoids. Inside of thylakoids, pigment molecules are bound and are able to do all of the work of the photosynthetic pathways. In prokaryotes, there are no chloroplasts, so photosynthetic pigments are instead embedded directly into the internal membrane systems. So that's always going to be internal to a cell wall, whether cells are gram-positive or negative, it doesn't matter. They all have a cell membrane that's going to be internal to that peptidoglycan layer. They might not all necessarily be lined up in a nice polite line in a simple cell membrane, but in fact, even though there are no closed off or compartmentalized areas within the cell membrane can invaginate to increase some surface area, and we'll see some examples of that coming up here. At the crux of photosynthesis is the existence of pigments, and only organisms that have pigmentation can perform this process. Chlorophylls are also known as bacteria chlorophylls, and anoxygenic organisms are required. Chlorophyll A is going to be the pigment that we're probably the most familiar with in higher plants, algae, and cyanobacteria. It's a green pigment and is preferentially going to absorb blue and red light. Even though chlorophyll is the one that we talk about the most, there are different pigments that will absorb wavelengths of light that are different than what chlorophyll will primarily like to absorb. And you can see here that the basic structure of the pigment has a hydrophobic portion that's going to allow it to insert into the membrane, and then the other portion that's going to stick out. And you might be saying, well, why would anybody ever want to have pigments that might not be as good as chlorophyll? Because if most of the world uses chlorophyll, isn't it the best? Well, there's also the ecological impact. If everybody is always using the exact same pigmentation in order to perform their photosynthetic processes, you're not getting as much out of that light as you possibly could. So imagine if some organisms will use pigments that absorb certain wavelengths, other organisms can use different pigments that absorb different wavelengths so that they're not competing directly with one another for the same source. That diversity can allow different organisms to coexist with one another and still both be able to support themselves. Pigment molecules tend to be assembled into complexes that are going to contain anywhere from 50 to 300 pigment molecules, and there is some degree of structure to them. You'll see that within the complexes, there are small numbers of pigment molecules that form what are called reaction centers, and you can see in this diagram here, they're designated by that RC and that darker pigmentation here. We also have antenna pigments that are going to help to catch and concentrate light into these reaction centers, which will really focus the process. If not for these antenna pigments, the low light intensities that are found in some areas of nature would not be enough for many photosynthetic processes to actually occur. So think of it as an amplification. If you're taking it and amplifying it or concentrating it down, the reaction centers will be able to do their work better than they would without the aid of that focusing agent. In some photosynthetic organisms, you'll see the presence of inclusion bodies, and you may recall that term from earlier on when we were talking about the basic anatomy of prokaryotic cells. Chlorosomes are a type of these inclusion bodies that will function as a giant antenna that will help to harvest and concentrate the light, like we've just discussed. And they'll have their pigmentation inside of the chlorosome structure. So you can kind of see in this cutaway here of some electron microscopy that there are these big areas near the surface of that cell membrane that look like they are partitioned from the rest of the cell. And these are those chlorosomes. You can see that they are conjugated to the cell membrane over here and will allow for that transfer of the light energy to reaction centers via the pigments, which are then going to be embedded inside of the cell's cytoplasmic membrane. So you can see here the reaction centers are located within the cell's membrane, and we have the big chlorosome with all of its pigments inside ready to do that work. Chlorophylls are required for photosynthesis, but there are a lot of accessory pigments that will additionally contribute to the process. Things like carotenoids are going to be yellow, brown, green, or red in color, and they're found widely in photosynthetic organisms from all areas. They're not necessarily going to be contributing to ATP synthesis like chlorophylls do, but they can act as antenna pigments and help to, again, concentrate that light to bolster the process of photosynthesis in the organisms that they exist in. Phycobilioproteins are red or blue pigments, and they can actually help to absorb lights at different wavelengths. A bunch of different proteins called phycourethrin, phycocyanin, and allophycocyanin can arrange in these aggregate formations to be able to, again, grab light from the environment, concentrate it down, and allow for energy to be transferred over to reaction centers where that work can occur. So you'll see in a bit more detail here that in our photosystem reactions, we have chlorophyll A, which is P680 of the photosystem 2 complex, is going to split water and take its electrons. The electrons are going to travel through a couple of different intermediates following the excitation, which will eventually donate it to P700 in photosystem 1. With more light being input, there's more excitation and more transfer of electrons along, which eventually results in that production of NADPH. And because this is a non-cyclical example of photophosphorylation, we do not see those electrons being returned to the beginning and cycled back through. The NADPH and ATP that get generated from the light reactions are eventually used to fix carbon dioxide to make sugars. This was first identified by Melvin Calvin. Ribulose bisphosphate carboxylase, or Rubisco, is the enzyme that's responsible for this process. You'll see here in this diagram that when we add carbon dioxide and ribulose bisphosphate, there is the eventual formation of 2-phosphoglyceric acid molecules. This process happens in bacteria inside of structures called carboxysomes. These are a type of inclusion body in only some prokaryotic cells that contain tightly packed arrays of Rubisco enzyme. And you can see some of those shown here in this diagram. These little clusters are those carboxysomes that we just mentioned. You can see here that some of the sugars that are produced from those processes can eventually be introduced into the Calvin cycle and used to form fructose 6-phosphate, which is a precursor that can then be used to form many different kinds of sugars through biosynthesis reactions or anabolic pathways. And you can see that product right here. When we look at anabolic pathways in general, there are typically 4 steps that need to be met. We have to convert a carbon source into precursor molecules that can then be used to make larger ones. We have to synthesize monomers and other building blocks into larger molecules. From there, we can synthesize macromolecules, and then we can assemble macromolecules into larger cellular structures. To do this, we actually have to use up quite a bit of energy and electrons from various sources, and this is going to prepare what's called reducing power. The reducing power is required because anabolism always involves adding electrons to compounds versus removing them, right? When we talked about catabolism, we always talked about things giving their electrons away. When we talk about anabolism, we're talking about things gaining electrons. So this is a great diagram just showing the different groups of organisms and how they actually get to the macromolecule or the storage molecule in some cases from smaller ones. You can see that depending on the energy source, the carbon source, or the electron source, it doesn't matter, but all of these are going to have their own pathways to get to this state where they can start to create large molecules from smaller precursors. At its core, anabolic pathways use energy to build larger molecules. Macromolecules are going to combine in many ways to form large complex cellular structures. This process can sometimes occur spontaneously in a mechanism called self assembly. Macromolecules have their own information needed to form larger ones, and sometimes it's just a matter of molecules being more stable when they're bound to others than not. So you can see in this example here, we've got some very small molecules of organic and inorganic origins coming together to form precursor metabolites that then get bound together and eventually form larger and larger complexes until we're very much up at the level of the cell, forming cellular structures, forming membranes, forming flagella, forming large proteins. It doesn't matter, but the level of organization of the cells is all based on that initial step of forming molecules from smaller components. When we consider biosynthesis in general, there are 6 major principles that apply. We have large molecules that are made up of smaller ones. There are a limited number of monomers that can make up macromolecules. This is going to save on genetic storage capacity, things like raw materials, and also energy. You can't have a billion different types of amino acids. Instead, we only have 20, and we can do an awful lot with just those, for example. Many enzymes are also amphibolic, meaning that a lot of enzymes that are implied in catabolic reactions are also involved in anabolic reactions in certain steps. So there is a degree of reuse so that we're not having a specific set of enzymes that are only tailored to one job in one pathway. In that same vein, though, other enzymes are going to either be strictly catabolic or anabolic, and that's going to allow for strict regulation of pathways that are going to be sharing intermediates. We talked a bit last time about how intermediates from one pathway can feed into parts of another and how it's all very interconnected, but making sure that those are isolated so that we're not having accidental conversion of things into others that we don't necessarily need is important to maintain the efficacy and the efficiency of the cells as they're undergoing these processes. Number 4 here says that anabolic pathways are irreversible, which does contribute to the efficiency of biosynthesis. If you were in the process of forming glucose and then 9 steps in, you decided to turn it around and start going the other way, or something interrupts that process and starts to reverse it, it would be very difficult to ever get anything done. So there is some degree of, again, that isolation that allows for those processes to be separated and not interfered with regularly. This is further achieved through compartmentation. That's going to physically separate anabolic and catabolic pathways from one another so that if the intermediates are similar in both, they're not going to accidentally be used for one versus the other. They're going to be funneled into the correct pathway and used where they're supposed to be used. And finally, anabolic and catabolic pathways are going to use different cofactors. Cofactors like NADH are pretty much strictly catabolic, whereas NADPH is going to be used in anabolic reactions. Now, we did touch on this a little bit last time about how, for example, deamination reactions can be used to produce intermediates that are part of the TCA cycle, but also glycolysis. This diagram serves to show some of that interconnectedness and how different molecules can feed into different pathways and aid in the production of monomers that can then go into alternative pathways and be used there. This really helps with adaptation because let's say an organism really enjoys eating sugar as its primary source, but one day sugar's not available and it has to switch over to something like protein. That's not to say that if that happens that a microorganism can't use glycolysis anymore to still get its ATP, because from that protein source it can still use some of those molecules through the breakdown of those proteins to feed into the glycolytic pathway and still be able to perform what they need to in their native metabolic pathways that they're most comfortable using. And this diagram is great evidence to show that no matter what the nutrient source is, metabolism can still happen. Gluconeogenesis is something that we've mentioned in passing a couple of times in these conversations, and this is a process that is essentially reverse glycolysis. We're going to produce glucose from precursors. It's going to share a lot of enzymes with the Embden-Meyerhof pathway, but it's more or less going to be reverse glycolysis, where we start with pyruvate and we work our way backwards all the way up to glucose. This process also produces other types of intermediates that can be used to make other sugars, like mannose 6-phosphate, which is really important for bacteria. We've been talking a lot about carbon dioxide fixation, but a lot of biosynthetic pathways can run in prokaryotes from 2 and 3 carbon molecules, like acetate. The glyoxylate cycle is yet another mechanism that microorganisms can take different types of molecules, so not necessarily the 6 carbon or the carbon dioxide, and still fulfill their biosynthetic requirements. You'll see here at a quick glance that many intermediates of the TCA cycle are used in this process, but the enzymes are very different. We skip a whole bunch of the TCA cycle here to produce glyoxylate instead. Now, if you're wondering how much I'm going to be asking about the specifics of these pathways on an exam, you're not required to memorize all of the different intermediates in these pathways or be able to draw them in any way, shape, or form. I may ask you to differentiate between them. I might ask you, for example, which one of these pathways uses a 2-carbon precursor versus a 6-carbon precursor or carbon dioxide. So you should be familiar with them, but you don't need to know all of the little itty-bitty details. One really important type of biosynthesis is the formation of peptidoglycan, which is going to make up the entire cell wall of most prokaryotic bacteria. The peptidoglycan structure is primarily NAG and NAM subunits, which are sugars, as well as some proteins that are going to be cross-linked. This process occurs by a synthesis complex that's going to be embedded in the membranes of cells, as well as in the cytoplasm and the periplasmic space. And this is a very complex process, so there are many components that are involved in it. In the final stages of assembly, we're also going to require transpeptidation, which is going to form peptide crosslinks or little protein crosslinks between peptidoglycan chains, and that's going to be the final step to kind of tie everything together, literally. Some of the chains are going to have direct amino acid interaction, while others are going to form an interbridge, and we did see this a long time ago in a little bit of detail, but we'll revisit it here in a new context. So you can see here in these transpeptidation reactions, all that's happening is these subunits that have been built of peptidoglycan are being stuck together. So we're having a reaction here occurring where this molecule is going to be joined up to the adjacent one by a linkage. So you see here, after the reaction proceeds, there is a link that is going to form, holding those two different pieces of scaffolding together. The same example here in a different species can also occur between different subunits, but still the same idea, where we are going to have a reaction that is going to form a linkage. And this is the example here of the interbridge, where we have several different amino acids that are forming a bridge versus just the unification of one. So this is going to be more tightly knit. This is going to be a little bit further apart. This diagram also shows penicillins here. This is not important to our current discussion, but penicillin does actually inhibit peptidoglycan synthesis. That is the method by which this particular type of antibiotic works. So if you're being treated with penicillin, this process is what that medication is actually going to target. And what you end up with as a result are cell walls that have no structural integrity, which will obviously result in the death of a cell. Now you might be saying, well, when do we need to make peptidoglycan? The cell wall exists once it's made, doesn't it? Well, cells are required to grow and divide, and as cells do that, you'll need to prepare more peptidoglycan to support that. Addition of peptidoglycan to the cell wall is very well controlled, and at first the cell wall actually has to be partially degraded, just enough to open up some regions that will accept new peptidoglycan that can be added onto. This is facilitated by autolysins that are going to go in and slightly degrade that peptidoglycan layer by breaking those polysaccharide chains and those peptide crosslinks that we just talked about. Cell wall synthesis can occur in one of two ways depending on the shape of the cell. In cocci, or our sphere-shaped cells, synthesis will occur at the site of septum formation. And that septum, again, is that portion of the cell that's going to come together as the two new cells are splitting off from one another. And in bacilli, it'll occur there, but also at other sites scattered in a cylindrical pattern across the rest of the cell as well. So elongation in a bacillus is going to occur more broadly than it would in a single sphere-shaped cell because they're smaller and they don't necessarily need as much growth or doubling of the cell wall material as you would need in a bacillus. So these two diagrams serve very well to show us exactly that. You can see in the coccus, in the cell wall, this blue region is that area of degradation and addition of new peptidoglycan. That septal region over time will be added to and will start to grow down into that cytoplasmic region and eventually come together and these two will eventually split off to become two new cells. In bacilli, you can see that that addition, the blue portion, is going to occur all throughout the cell, but in addition to the septum. So those cells are going to elongate differently because there's more required for those larger cells. Amino acid synthesis is also extremely important, not only for the peptidoglycan layer, but for many different cellular processes that involve enzymes. Amino acids are going to have three components, if you recall. We have our carboxyl group, the amino group, and the side chain. Carbon skeletons are going to be used as precursors in their formation. And there also has to be a way to incorporate nitrogen into the cell, which is obviously something that is found in every single kind of amino acid that gets produced. In addition, other amino acids require other atoms like sulfur to be incorporated. Many amino acids are formed through what are called anaplerotic reactions, which will use intermediates from glycolysis to generate something like oxaloacetate, and that can be done from using phosphoenol pyruvate or pyruvate itself. So you can see here that from either oxaloacetate or phosphoenol pyruvate, you can produce quite a few different types of amino acids just from a couple of different conversions. So you can pull those intermediates that also exist from other metabolic pathways to shuttle off and make new types of precursors that might be required to build up some enzymes or peptidoglycan or whatever protein might be required for the cell. We could get into the discussion about the synthesis of purines, pyrimidines, and nucleotides, which would require the assimilation of phosphorus, but that is a very large set of pathways that we won't be discussing here. But you should know that phosphorus is important in phospholipid formation, which is the primary component of our cell membranes, as well as a lot of proteins and coenzymes. And there are three mechanisms that we can assimilate phosphorus, which are photophosphorylation, oxidative phosphorylation, and substrate level phosphorylation. When we look at lipid synthesis, this is yet another process that is going to use intermediates from other pathways to function. Fatty acids are made from the addition of acetyl groups from molecules like acetyl-CoA, which you may know is some of the precursors that go into the TCA cycle, and also malonyl-CoA. Fatty acid synthase is the big enzyme that's involved in this process, and what will happen here is that that chain length is going to grow until it's going to be added onto that glycerol backbone or onto another compound. And again, in terms of these other processes that we've discussed towards the end of this lecture, you're not responsible for knowing any of the intermediates, but just kind of be familiar with them in general. That'll just about do it for now. Thank you very much, as always, for your attention. And if you do have any questions, you can always contact me at laura.r@nipissingU.ca. Thanks very much and have a great day.